just now getting our very first views of the bottom.
Yeah, excellent. So we do see a little bit of water motion here, and we do see the ridge coming into view here for the very first time. And again, it's very exciting. This is the very, very first time we're surveying this area. This has never been sampled, even with dredges or anything like that.
Keep coming down
up until yesterday, there were no maps of this area, and so these are just yesterday we finished mapping it, and now we're gonna be diving on it and sharing with you this very, very exciting moment.
Come down just maybe 10 meters.
So during our ascent, it looked like there was a lot of pelagic marine debris and detritus that was raining out, or raining, kind of dropping in the water column. But the surface of the seamount seems to be very lightly sedimented.
Bridge, ROV nav.
So that's good news for geological observations. It means we're going to be able to see a lot more rocks and a lot less sediment. And we're going to be able to make some observations about the lava flow shapes, and maybe be able to observe some primary lava flow textures. And when I say primary, I mean that the texture on top of the lava rocks and lava flows that we see might give us an indication of which direction they were flowing, how fast they were flowing, and what kind of interactions the lava flow encountered on its way out. So this is kind of the story that geologists always try and put together. We try and make some observations in order to tell the story and the life cycle of a rock.
So yeah, we just touched down. Current depth is 1,290 meters.
Stand by for me. We're just getting settled.
And there's already a whole bunch of rocks there. So yeah, that's one of the things we'll be looking at, is to collect this sample.
So it's actually a great thing that we are already starting to see some loose rocks and debris at the very bottom of these seamounts. So what happens to these seamounts is, as they get older and older and older, a precipitate that is made out of manganese and other heavy elements starts to grow over the surface of the rocks. This manganese crust that you'll hear us talking about over and over as the dive progresses. So this heavy black coating precipitates or grows on top of these rocks as the rocks sit, and the longer that the rocks are on the surface and sit, the more manganese crust can precipitate on top of them, eventually cementing the entire surface together. Loose rock or intact rock, it doesn't matter. It grows on top of it, cementing it together and making it almost impossible to acquire a geological sample. So the fact that we see some loose geological samples that the ROV can collect right away in the dive is boding well for a successful geological collection. So the reason that we're going to be collecting rocks as well as biological samples on this dive is so scientists can then process them in their labs after we get the rocks back to shore. And some of the things that geologists are going to do with these rock samples is the very first thing that we want to do is we want to get the age of the rock. And so geologists will take this rock and they're going to use a certain part of chemistry called radioactive decay. So the amount of two different types of the same kind of element will be analyzed. And it looks like we have our first animal. What is that, Daniel?
So we have a halosaur, so a lizard fish. And yeah, there's at least 3 genera we have now here from Hawaii. One that we just found a couple of days ago. So distinguishing, we'll have to look on the head. But yeah, Bruce Mundy, who is joining us at IRC at Ford Island, maybe can give us his first impression here.
Okay. Hey, you can come out. I'm just.
So I really need to see the scales on the head before I can say what it might be. There's been a lot of discussion in emails about this, and it's whether or not the scales go up over the snout or not.
Maybe 5 meters.
So from this view, we can't see that.
Yeah.
Seeing in would be really great.
Yeah.
We had some communication with one of the world experts on this group, Ken Sulak, who originally suggested the 3rd genus here in the islands, but he's rethought that in the last couple of days. and now we think there are only 2 genera,
coming at me.
with at least 3 species, though, in the 2 genera here. And these scales on the head and along the body behind the head toward the large fin that goes along the bottom of the tail, the number of scales there are important characteristics for identifying these. So
it's hard to track.
getting a close view of the head, the shrimp's great too, by the way. Getting a close view of the head and then also of the front part of the body would be really good.
It's going the other way.
Excellent, Bruce. Yeah, that was Bruce Mundy from the Pacific Islands Fisheries Science Center, who's been joining us on every one of these dives, and one of the experts on fish taxonomy here in this area as we're taking a look at that fish. So yeah, the fish we saw, that is in the family Halosauridae, so they're halo and saurs. They're basically a composite word meaning sea lizard or ocean lizard. So they're the lizard fishes, and yeah, hopefully we'll get a better image of them here in a little bit.
Gotcha.
And Mary Wakeston, who is joining us from Texas A&M University, one of our experts on arthropods and different groups of arthropods, has just identified that shrimp as probably one in the family Aristeidae.
You feel free to zoom. I'll try to keep
Andrea Quattrini, who's in the chat room, but I don't think it's on the conference call, thinks that this is the genus Aldrovandia instead of Halosauropsis, but we'll find out here in a moment. Unfortunately, it's tilted a little bit, so we can't see the top of the head, but there may be enough that we can see to get a better idea. It's rather pale. It's interesting. It's more pale than most of them that we see.
Okay, come back in.
Excellent. Thank you, Bruce. Yeah, so we'll try to get a good view for you on the top of the head and take a look at the scales.
Lasers are clear.
Okay, from what I'm seeing now, it looks like there are not scales on the front of the head before the eye, which would confirm Andrea's identification as Aldrovandia. Maybe she'll chime in the chat room. I don't see scales on the snout, so Aldrovandia. Aldrovandia was discovered here in the islands back in the Albatross expedition at the turn of the 19th and 20th centuries, with 2 species reported. The other genus that's here in this group, Halosaurus, was newly discovered for the region in the last year's Okeanos exploration dives. So we had a new discovery in this family from last year. This does look like Aldrovandia, and it'll take a little work to figure out which of the 2 species it is.
Yeah, thank you, Bruce. And yeah, Bruce has been working a lot with different fish taxonomists from around the world, and yeah, he recently shared a very interesting fact. So last year we did about 18 dives in this area, and from those dives we imaged close to 200 fish, and about a quarter of those were new records for the region. So very interesting. And so there's a second fish. Maybe we can take a quick look in the background.
Whenever you're clear, video.
Video's clear.
Okay.
Now that looks like it might be a little synaphobranchid eel. It's hard to tell, but it looks more like a true eel. These Aldrovandia are what they call spiny eels, in addition to the sea lizards, because they have a dorsal fin on the back with a little spine.
Below that white spot on the right.
I suspect that fish in the back that we might see could be
good spot.
I can't remember what they call them now. Cutthroat eel, or there's another name maybe Andrea remembers.
That's a partial.
Copy. Just hold there.
Thank you, Bruce.
His head's at the top.
Okay, go ahead.
Yeah, and this is something like we've seen on a few of these dives. Is in those places we find a lot of fish. We don't find as many corals, and vice versa.
Oh.
Now that I get a closer look at this, it looks like a juvenile. I'm wondering if it might be a juvenile cutthroat eel in the genus Synaphobranchus. I don't know if we can get more video of that.
Yeah, he's a little skittish, Bruce. We'll try to get that.
Sounds like they may want more.
Yeah, understand. Not all fish are as cooperative as that Aldrovandia.
And we're just going through a quick personnel change here with our ROV team, so we should be on the move here in a second.
So the little eel failed its screen test. The Aldrovania
Come
gets
down
the
as I
part.
push up, unless we want something from right here.
Excellent.
Watch lead, how's this?
And now from the two we imaged last year, Bruce, one we had like tentatively ID'd as Phylacra, and the other one just as a unknown species.
Pilot, as you were
It looks
changing out
to me
there,
more like the
scientists were
Phylacra.
talking about trying to get another shot of that little guy.
Another shot of the smaller fish.
Yeah, I don't know if it's still okay.
Yeah,
but
I'm
that's
not
what
as
they're
familiar
talking
with these as I should
It sort of
be
went down below
for
that
quick
big rock.
identification.
Down below the
So
big
I'll
rock
need
guy.
to do more work, and we'll communicate with this with Dr. Kenneth Sulak, who's in Florida. He may be participating remotely, but he's not online here. He's the world expert on this group of fish. So we'll find out what he thinks about the fish in this video.
Excellent. Thank you, Bruce.
Okay.
Hey, Pilot, this is Watch 2. So I think you were asking about some of these samples down here. It looks like there's a wealth to pick from. So if you guys are, whenever you guys are ready to try and make a collection, it looks like we've got a lot of very angular, kind of boxy type shapes down here. Probably go for one of those.
Okay. Let me turn on the lasers here so you can have something to first gauge the size and something you can use to direct me to the one that looks best to you.
Yeah, copy that, Pilot. Thank you. Yep, see the lasers.
And we'll just, let me tilt up so you get a better view of everything. And
See some legs off to the left.
And
of the rock.
Let's see. So yeah, so you just, that guy that the lasers are just below, or above right now?
Just above?
Yep, so there's two actually that look pretty good. There's one that the lasers are on right now that looks maybe a little too large, but it looks like a good sample, and then there's a couple within that are also very good candidates. So if we...
All right, then I'll come in and settle here.
Great, copy that, thanks, Pilot.
And if you could come down a little for me, co-pilot.
Yeah, come down a little.
Thanks. Good. Pushed in here. Okay. Tilt down. Let's see, that's good for altitude. Want to spin us up?
Which view on? Ramping up.
So if you're just joining us, our ROV team is currently setting up to make our very first geological collection of the dive, and the very first geological collection that's ever been sampled from this seamount.
Sweet. Thank you.
It's a rare site that on seamounts that are apparently about 80 to 90 million years old that we touch down on a surface that has loose rocks that we can actually grab, and this is due to a inorganic precipitate called a manganese crust. You'll hear us mentioning this over and over. And so what happens is, as the rocks are sitting on the surface of the seamount, they start to grow this black coating on top of them called a manganese crust. And the longer that they're around, the thicker the manganese crust gets. And so eventually this manganese crust starts to cement the entire surface together. And so what we're looking at right now is a very nice collection of loose rocks, and not only are they loose, they're very angular, indicating that the relative surfaces of these are actually not that old. Now, that doesn't actually mean that the rock itself and the seamount we're diving on is not that old. It just means that these could be the products of a recent landslide, and the surfaces of these rocks are actually not that old. And this is of great interest to geologists. So we're looking for a loose sample, one. Two, we're looking for one that it has a lot of fresh material to work with. So the more fresh material that we've got to work with, the more laboratory analyses we can conduct on that rock. And so a couple of the things that geologists will look at is, first things first, they will slice the rock open using a saw, collect a thin section sample from it. And so from the thin section, we can then put that into a petrographic microscope. We can identify the minerals inside of them, the relative sizes of the minerals, and we can start to piece together a story of what happened to the magma while it was in the magma chamber and growing its very first crystals, all the way to when that magma possibly erupted as a lava flow and what happened as it cooled. So after the rock has been made into a thin section, we will then crumble the rock up into small pea-sized little gravel bits, and then once we have those gravel bits, we then powder those gravel bits into a fine-grained powder. We then analyze that powder for its chemical composition using a couple of different techniques in which we use basically a mass spectrometry of different methodologies to reveal the chemical compositions that make this rock up. And based on the chemical compositions that make this rock up, we then can even say a little bit more about that rock. You know, where did it form? How did it form? What kind of melt of material did it come from? And so after we have powdered the rock, as the third thing that geologists are going to try and attempt to look at is dating the rock using radiometric age dating. So this involves looking at the relative distribution, or the how much of two different types of the same element are inside of the chemical composition of the rock, and this tends to be two different kinds of argon, argon-39 and argon-40. So based on the relative abundance of argon-39 to argon-40, we can actually get a very precise age of the rock. And so for those of you that are seeing this rock being collected for the very first time, and you also see two red dots just on top of the rock right now, the width between those two dots is about 10 centimeters. So this rock has about a width of maybe approximately 15 to 20 centimeters on average. And it's very angular, which, like I said earlier, is going to kind of give us a relative idea of the degree of alteration that has taken place. So hopefully within this dark manganese coating that you see on the surface of the rock right now lies a very fresh core of nice volcanic rock for a geologist to analyze in labs later.
Excellent. Yeah, thank you very much, Jonathan, for that, putting that all into context. Just to let our shore-based scientists know, our chat is currently down, but we did see your the messages that you sent just previously. So yeah, after this collection is done, we'll try to image that coral that's to our right. But yeah, very exciting that we're just successful in collecting the first sample ever from this seamount that up until yesterday had not been mapped, and now we know what the what this whole feature looks like, and now we can see something on it. Looks like a sponge material, possibly. Several sponges that have this kind of attachment to the rocks.
Oh, look at that. I got the blade.
And so, yeah, these are the first images ever taken on the seamount, and the first sample ever taken from the seamount. So again, as of yesterday, this very tall seamount that's close to 13,000 feet tall had not been mapped. We knew that there was something in the area based on satellite altimetry. So satellite data, with satellite data, you can take very, very fine measurements of the height of the sea level. And interesting that the waters actually pile up on top of mountains. There's a slight increase in the elevation of sea surface height on top of seamounts, and conversely, there's a slight depression of the sea level on top of depressions or trenches. And so by taking very accurate measurements of the sea level height, you can try to understand what might be underneath it. But with that kind of technology, you can only resolve features that are about 1 kilometer in height. So it really gives you only a very coarse view of what might be underneath. And what we do to try to get a better understanding is multi-beam mapping. So there's an acoustic signal that is emitted by the ship. There's a so you basically make sound, and that bounces off the sea level and comes back to the ship, where sensors can measure that. And based on the time it takes between when you emitted that signal and when it comes back, you can figure out exactly how deep it is underneath you. And so that's what we've been doing on these last few days when we're not diving, is collecting high-resolution mapping data of the area. And in the process, have come across some very interesting features. At least 4 seamounts that we had not mapped before. One inside the Papahanaumokuakea, just west of Pioneer Bank Ridge, and 3 additionals here just on the periphery of the monument. The one that we're diving off about 20 miles west of Salmon Bank and about 20 miles south of the boundary. Yeah, thank you very much, pilot.
I don't like to cut in, but just to the right of where you photographed the hadrosaur, there's a gastropod mollusk on that big rock. It might be worth looking for him if you're on your way to the coral.
Excellent. Thank you, Marianne. I appreciate you pointing that out. Yes, as soon as we stood up, we're going to take a look at both.
You got a target for that sample, Nav? Yeah.
I believe to the right. So when we
She said it was
on the to our right, even further out of view, there was a big. There you go. There's a coral on there, and I think one of these rocks.
Well, there's that anemone up there. There's mushroom coral. There's a smaller coral branching one. Which one of these are you looking at? Sorry.
Mary, to see in view now what you were referring to?
And I've got a shrimp.
Yeah. Maybe we can start with the coral, and then Mary can guide us.
Okay. We'll start with the coral. Okay, video, you can start in.
So this is a pink coral.
No, it was a white coral. It's the one that somebody else spotted just a couple of minutes ago.
Oh, I saw the white whip coral. Is that what they're talking about?
Yeah. Yeah, there's another coral. We'll image here, and then, so yeah, this is what we're looking at is it's in a family of Corallidae. So these are the pink corals. Just to the bottom of it is actually a barnacle. Oh no, that's a polychaete tube worm.
Okay.
And on the pink coral there we have an ophiuroid. In the background we also saw a sponge just to our right, and that's going to be in the a sponge in the Furidae family, and look like Furia near Oka erecta, a common inhabitant of these kind of depths.
Okay. What's the target we're looking for?
I believe it's to our right.
I saw the whip white coral on the right.
Yeah.
We'll definitely get that for video. It looks really nice. But let's get this coral for science real quick. And there's another coral way off in the distance to the right there. I don't know if you see that wash lead in the top corner there.
Yeah. So whatever's the most efficient. If you want to get the anemone first, if you need to get that for video, we can.
Is this
before.
whip coral what we're looking for here?
No, it was a
okay.
tall coral that was on top of a rock, and I believe it's still to our right.
Still further to the right. Okay.
Yeah.
So that small branching one over there. Do you see it now? Small center screen. Or that whip now showed up bottom right.
The shrimp at lower left is probably the Metacarcinus.
Do we know? You can snap on this one if you want. Yeah. See if that's the one they're talking about. Is this it,
Yeah,
actually?
I believe. Yeah, that was a coral that we had spotted earlier.
Is that the one you want to go take a closer look at?
Yes.
Okay, video. Come on out.
Pilot, you're teetering on my view. Do you want me to rotate?
And then Mary, if you can help us,
Sure.
let us know where you spotted this gastropod.
It's further down from where you are.
Collins 217, Mike 217, or come on down.
Excellent. Thank
Right
you.
where you first came in.
Oh, so to our left now.
Do you still want this?
Yeah, let's take a look at him.
Okay.
So yeah, we do see some animals here in the area. We did image a couple of corals there too just a while ago. There was a pink coral, a mushroom coral, a couple of sponges. And so this is a bamboo coral.
Maximum zoom.
And just to let our shore-based scientists know, so yeah, we currently do not have the chat working.
Go ahead and pan down the stop.
So please call in if there's something that you need to relay to us.
Hold fast.
Okay, going to go set up for full species.
Excellent. Thank you, pilot.
Hold the shot.
So, pilot, if you need to get that anemone for video, let's get that, and then we'll try to make our way
Video's clear.
over to the
Okay.
towards the slope here.
Okay, so we can head for the anemone and then head up slope. So what you're saying is the anemone of any interest to you?
Not particularly, but yeah, if you need to get it for video, yeah, it's gonna.
Call. You can continue on up slope unless you want to chase that down.
Yeah, no, let's go ahead. I know what it is.
Okay, we'll continue on then.
Copy that. So again, yeah, current depth is 1,290 meters. We do see a few animals here in the area. So far we've seen a couple corals, some mushroom corals, and a few sponges, but this is kind of a flatter area that's going to be transitioning into about a 45-degree slope on average, and that's going to be until we reach the summit or the top portion of this seamount, which kind of flattens out to around 700 meters. So we'll be moving up along the crest of this kind of ridge, and in the process we'll be transversing an area where we expect to find
Sorry, just settling in the
lower concentrations of oxygen. So right now we're just at 1.5 milligrams per liter, and we'll see how that changes as we move up. It should be dropping down to values below.
Beautiful view from Sirius.
Dan, we have visitors from the Waikiki Aquarium here now, volunteers with the aquarium. Over.
Copy that. Yeah, so good morning. Welcome. Thank you so much for joining in to our visitors from the Waikiki Aquarium. You are watching live video feed that's being brought to you from the NOAA ship Okeanos Explorer that's currently spending about 3 weeks up in the northwestern Hawaiian Islands, part of the Papahanaumokuakea Marine National Monument. And these are images. We're close to a mile deep and about 1,250 miles away from you from Honolulu. And so you folks probably know a lot about Papahanaumokuakea, knowing that we have our tank there at the aquarium with some species that are found only here in the northwestern Hawaiian Islands. So yeah, welcome and thank you so much for joining us. So this is going to be a tube-dwelling anemone or a Ceryantherian.
It's one of the best views we've had of the tube and the anemone.
These are not true anemones. They're actually not as closely related as one might think. The difference here is that they will have multiple rings of tentacles, and these guys will have one shorter one, kind of a ring of very short tentacles close to the mouth, and then a lot longer tentacles on the periphery. And they will use those long tentacles to capture food, and then once that food is captured by mucus that basically lines these tentacles, it can then move towards that inner ring where those short tentacles will be used to manipulate it and move the food particles towards the mouth. This is a group that's not very well studied. Few information exists, and the taxonomy, so differentiating these two species is also very difficult. A lot of times the internal structures are used to differentiate the different species, genera, and even families, so you really need to sample, and then you need to do some dissections and take a look at the internal structures.
Alright, I'm coming up towards you, co-pilot. As I come in, just continue sidling left.
So yeah, that's a common theme with a lot of these groups. Is in certain groups, it's the internal structures, it's skeletal elements, things that are used to classify many of these organisms, and as a result, you really need the specimen to be able to even answer the most basic of questions, which is what species is that? So can we take a quick look at that tall, large coral to our left?
Of course. Just getting a established shot of the nice geology.
Yeah, that was great. So that was
this tall whip coral here?
The one in the foreground, yeah, I don't know if it's the same as before, but it seems to be the dominant species here.
Like the ones in the lasers?
So the one the lasers are on the base of, or this?
Yeah, the one in the foreground.
This guy here.
Yeah.
that really nice transition between what was actually a primary texture from the actual rock itself. Looked very dense, not very vesicular, normal of lava flows. It kind of looked like what would be a horizontal dike, which is what we call a sill.
Yeah, you can start in.
And then right on top of that, you could see the heavy manganese crust that I was talking about with a texture that geologists call botuloidal texture. So botuloidal means literally like clumps of grapes. So that globular texture of the manganese crust, that heavy manganese crust on the very top of the rock outcrop is it's a nice illustration of what I was referring to earlier when the manganese crust has completely cemented the surface altogether.
So yeah, this is a primnoid coral with an commensal ophiuroid on it, downward facing polyps, possibly in the genus Calyptrophora. We'll take a closer look as we zoom out here.
One for full species.
Okay.
And yeah, it looks somewhat similar to Calyptrophora wyvili.
You have this on mute? Hello?
I'm just going to tilt up real quick video.
Did someone...
Please check your conference phone.
Lasers.
So, Chris, I think that was you. I could barely hear you, but was that you calling in?
Okay.
No, sorry, we're just setting up right here. We're a little late setting up, and we were testing the microphone. Sorry.
Yep, well, we could hear you, but only faintly, so I can hear you a little bit better now.
Swimming in the back, or is that just dust particles? Flare.
Excellent. So yeah, that's Chris and Andrea who are joining us from the Exploration Command Center at the University of Hawaii. So yeah, for the next, or throughout this field season, there is a Exploration Command Center at the University of Hawaii where a really nice setup with a lot of screens that very similar to what we have here on the ship. And so it's a beautiful facility. And yeah, we're starting to see a nice moderate density here of corals. These are all primnoid corals. So we landed just off this ridge, and now as we are kind of coming on top of the ridge, we do see a nice moderate density of primnoid corals. All appear to be the same species here. Yeah, so maybe we can take a quick look on one of these unbranched corals, the taller one to our right, and then we can start our transit here.
Take a look at this and then start the transit. Copy that. Tilt down a little.
Fish on the left-hand quarter.
Oh, okay. You want to snap into that real quick before we get the coral? Tilting up. You can snap in.
And
Back tail. Got any more? Is that full? Go.
And so this one is, I believe, is new for, I don't see it in any one of the gallery shots from last year's expedition.
Do we want a closer shot, Saints?
I think this is probably good, yeah, if we can just go up and just pan on the body. But yeah, we got a beautiful image here.
For the body.
Hi, Dan. Hi, Jonathan. This is Andrea Quattrini. Can you hear me?
Yes, we can hear you, Andrea.
Hey, welcome.
Great. It's good to be finally connected here at the ECC. It's a great shot of a McRaran. I don't know if Bruce is online, if he may have an ID on it.
Nice video.
Yeah, and
Nice work.
Bruce was with us, so yeah, I'm sure he will either have seen that or will soon see it.
Lift up and tilt down to get the light out of our eyes. We can.
Yeah, the rat-tail family Macraridae. It's a very diverse deep-sea family of fishes, and a lot of them are actually fairly difficult to ID using only images. But I know Bruce has extensive knowledge in the area, so I'm sure he'll be able to help.
Yeah, thanks, Andrea. So yeah, he is with us. So now we're looking at an unbranched bamboo coral. And you can kind of see there why this group is called bamboo coral. So you see those alternating lighter black bands and then the whiter bands, and then we have a commensal squat lobster on just perched on the branch of this bamboo coral. And there's a second one towards the top. Mary, is this also the, well, we have them as the Europtychus?
Pulling for the player.
Okay.
Hi, Dan. I think the event log again is out, but Chris and I were just talking here that it looks like a Europtychus.
Yeah, excellent. So, yeah. So a very, very common kind of symbiosis or living together of organisms is that you see squat lobster kind of perched on top of either sponges or corals, kind of using these corals as platforms to get off the bottom of the seafloor. We have a very low current flow, and as you get further off the bottom, there's more flow up there, and so squat lobsters will kind of use these corals as platform.
Creatures aboard.
And yeah, it's also a diverse group. The squat lobsters, they're not, or they're different from the true lobsters in that their body is kind of flattened like a pancake in the back to belly direction and dorsoventral direction. There's also some species that are harvested commercially and used as food. Langostino, for example, if you might have seen it on menus, that is a squat lobster.
Lagostinos is a type of squat lobster?
Yep.
Oh, they're so good.
Bad pilot.
Excellent. Yeah, and so Mary, yeah, thank you. Yeah, we just got the chat back up and running again, and so yeah, Mary confirmed our tentative initial ID that there's a.
You've got a on board.
That this is a Europtychus squat lobster in that genus. Looks like we have a crinoid there at the top, a unbranched or unstalked crinoid, also known as feather stars. And that is also a common symbiosis. And so, yeah, a lot of these corals really are prime habitat for a lot of other species. As I mentioned, if you were to measure current flow very close to the bottom, it would be negligible to none. And then as you go further up the substrate, you would have an increased current flow. We call this the benthic boundary layer. There's this kind of boundary right next to the substrate where there's almost no current. And then as you go up away from the substrate, there's a kind of exponential increase of current flow as you get away from the substrate. And so a lot of things need to get off the bottom so they can have more flow over their tissues. So they can, because a lot of these things are suspension feeders, feeding on tiny particles that are in the water column. And so getting off the bottom is beneficial.
Great. Thanks, Roland.
So yeah, we're already here seeing a nice aggregation of animals. We are right
All right, guys, do we want to think about starting up slope?
Yes, please.
Yeah. Let's look at a shipment of sorts.
Maybe quick snap zoom on the one that's to the bottom right. Bushy.
We're getting a video change. I don't know how quickly we can zoom. Unless she gets her headset on, maybe. Sorry to race you in, video. Could you just snap in on this coral? Thank you.
So yeah, that's a chrysogorgid coral, and that's likely a small Chrysogorgia stellata with once again one of these squat lobster there, that red spot that you see in the center, and that's exactly what I was talking about before.
Thank you. Shot of the base.
So a lot of these squat lobsters, as well as crinoids and other things, just using the corals as habitat to get into places where they can feed more efficiently. So yeah, this is in a different group, that small coral that we see in the center, to the one that's to the right. That's a chrysogorgid coral. They have kind of elongate polyps, narrow elongate polyps, and then the primnoid corals, which seem to be the dominant group right here, are the other ones we see here in our area. We also did see a bamboo coral that's in another group. So yeah, we do see nice moderate density of animals right here, and also a different species here. Maybe a quick zoom on this this purple animal.
15 degrees.
Purple.
Right to the bottom.
Slowing down for it.
Okay, 30 meters.
I'll call it in now.
Nice.
Okay, video, you can start in. I'll tilt.
Roger.
So yeah, that looks like Victor Gorgia nutting eye, one of the most conspicuous inhabitants here because of its
Bridge. This
coloration.
Yeah, I'd like to request a ship move 30 meters, bearing 015 at a speed of 0.2 knots.
And
good copy, Bridge.
There in the background, you see an ophiuroid or bitter star just that jumped off things. Another animal, and Chris Kelly from the University of Hawaii had just noted that a lot of these primnoid corals that we mentioned a while ago, those kind of that look like candelabrum, are likely in the genus Narella. The primnoid corals overall, it's a somewhat difficult group. There's not many people working on it, so again, for those viewers and for our young listeners and viewers, there's a lot of these groups that desperately need some passionate individuals to be working on them.
How fast would that move? 2 or 3?
0.2.
2 tenths, thanks.
So if you have an interest for the ocean and exploration, there's a lot of work to be done. And there was this, yeah, and so yeah, next to that coral we just imaged was one of these long-legged shrimp, the bright red, and that's in the genus Nematocarcinus. And yeah, let's take a quick look. It looks like a Sacocalix. Let's take a quick, I'm sure Chris will appreciate a quick look.
Yeah, I'm a little stretched out, so we'll do it from here. Go ahead.
Okay.
Video.
Yep, and that's exactly what it is. So that's going to be a glass sponge in the genus Sacocalix.
I am coming in towards it still. Just so you know, video.
Roger that.
And again, kind of illustrating the same point I was making about earlier, is that getting off the substrate is of uttermost importance to these organisms. So yeah, you see a lot of these modifications, things like growing on stalks. So the top there is where they would feed. So sponges, they have this very porous body plan, and water will flow through these pores, and they actually have specialized cells that will help pump water through these cells. They're called canocytes. And so yeah, but again, getting off the bottom through these stalks is what these things need to feed more efficiently. And Andrea had noted a skeleton shrimp, the coprolid. I don't.
At the bottom on the stalk.
Oh, you want to take my camera, co-pilot?
Absolutely.
Tilt down.
Tilting down. Do you want to see the base?
No, that's
There's a coral.
is
I
that
think, on
skeleton shrimp?
Yeah,
there?
right. Yeah, excellent. So that's going to be a different.
And do me a favor. Is this full zoom?
Yep, it is.
Okay, can you come wide real quick?
Sure thing.
And I just saw something kind of funny in there. Let me just lift up and get a little closer.
So
you
in the
can see it.
background there, that's another glass sponge or a hexactinelic sponge.
Yeah, you have more slack.
And
now.
that looks like it's going to be in the family Furidae. And it's this sponge that we've been, it's in a genus Furia, and the closest is Oca erecta. Also very conspicuous species here in these kind of depths.
Again.
Mm-hmm.
In this hole, I saw, I think from this view we'll see a little better, I saw the smallest little polyp, it looked like. Can't
Do you want me to?
tell. It's similar color to the sponge itself. Do you see that little
Yeah.
crispy polyp in there? You got any more zoom video?
Let's see.
Yeah. You see that? Science. Is that part of the
Yeah.
sponge or is that something else?
No, it's something else. It could be something living inside, and it's just obscured. But yeah, it's hard to see from here.
Cool. Okay. There's a few of them in there, all sort of lining the inside. That was interesting.
Yeah.
It's cool for you to see inside like that.
Oh yeah, Chris, are you seeing this? Is that one of those anemones there? You can actually kind of faintly see it. So last year in another glass sponge in the La Froyella, we saw this association commonly that there's
Ready when you are.
a lot of these anemones apparently growing right inside, and that looked
Good spotting.
very much like tentacles from a hexacoral, or which the anemones belong to. They have polyps that are unbranched, unlike the octocorals, which have the tentacles itself are branched. And then they also have tentacles in multiples of 6, and unlike the octocorals, which have 8. And we'll see examples of both of those groups.
Pilots, looks like the ship has finished her move. We got a couple more minutes of sway.
Can we take a quick look? No, actually, those are dead sponges. We're good.
You good?
Yep, thank you.
You see this interesting shape? Actually, I don't know if it's a piece of wood or a weird mold in the rock or what. I haven't seen that kind of shape in the rock yet.
Yeah, definitely. Can we maybe zoom in on that a little bit?
Of course.
It looks like a rock surface,
Yeah.
but it has that lineation along its long axis. It's kind of interesting.
Yeah, sorry. Just trying to get a quick toe hold of it.
Oh, pilot, you have a fault. Do you want me to adjust?
Not now.
Got it.
Video, come on in.
Wow, what is that?
Is that a piece of wood of sorts?
Yeah, it looks.
Not really sure how to describe that.
Do you want to see? I mean, ship move stop.
Yeah, should
Curious to see if it's loose or a part of the surface.
Copy that.
Thanks, Brian. So the only explanation I have for this is that this is a texture generated by the manganese laminations and incrustations that are kind of laminating layers successively on top of something, and then the shape. I'm not sure why it has that shape.
Could we zoom on the bottom of it?
Weird. Good eye, though. That's
Coming in on the bottom. It's really folded and leaf-like there.
Oh.
It's really strange.
it is very strange. I have no explanation for this. This is such a weird object. I'm pretty convinced that it's composed of manganese. I'm just not sure why it had taken the shape that it has.
Yeah.
Obviously, it doesn't belong there, so it's a piece from something else.
Yeah,
Yeah,
so Diva, yeah,
Oh,
said
whale
that
bone.
she's pretty convinced that this is a manganese-crusted whale bone.
Wow.
Oh, well, that's why it doesn't have a geological explanation. I'm off the hook.
That's really cool.
Yeah, but the fact that it's heavily crusted gives you an idea of how long it's been there. I mentioned that manganese crust is a very, very slow process of accumulating, and yeah, you could see it definitely having a very thick layer of manganese crust on there.
Do you want to see if it's crusted to the wall? I'm sure we don't want to collect it.
No, we're good. Thank you. Yeah, that's enough.
All right.
Appreciate it. Yeah.
Thanks, video.
Sure thing.
So yeah, thank you, Diva, for sharing that and providing that concept. So yeah, this is the very nice kind of model that we have here on the Okeanos Explorer, is that you have a whole group of shore-based scientists who are also watching the live video feed in real time and connected to us via conference line and helping to provide context and interpreting some of the observations and then guide the observations. So it's a very neat way, this unique what is called telepresence communications technology. So the video stream is broadcast live so that both scientists and the general public can enjoy these images at the same time that we're seeing them on the ship.
Snap in.
Hi, Daniel. This is Andrea Quattrini. You're breaking up quite a bit on shore, so just curious how we're coming in, if you can hear us loud and clear.
We hear you loud and clear.
That's a very bright color.
And we're also taking down the second video stream, so video should improve in quality over the next couple minutes, I'm sure. But
It's a nice image of a crinoid and an ophiuroid.
Yeah.
Bump, 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 bump. All right, thanks.
Yeah.
Sorry about the bumping at the end there.
That's all right.
And there's another mushroom coral there, that red, also very conspicuous species here at these depths. And yeah, that looked like Kleptometra, this yellow one, and Chris suggests Kleptometra lateralis, one of the more common crinoids in some places. You see very dense aggregations of them. Depth is 1,264 meters. Dissolved oxygen level is just around 1.5 milligrams per liter. So throughout the course of the dive, we should see that dip below 1, and around like 0.7, and we'll see how that shapes the communities. So yeah, we see a nice moderate density of, most of them are permnoid corals.
Okay.
And there we see our first sea cucumber of the dive.
His like arms or tentacles or whatever those are much longer than, usually like little tiny spikes. They're so long.
Get a full zoom here.
So yeah, that's in a family. That's the Dematidae. So yeah, you have those projections of the body, which characterizes that family. So the Dematidae holothorians, and also that translucent coloration of the skin. That's also a kind of common adaptation that you see in the deep sea as you start seeing a lot more gelatinous body forms, less color. There's a lot less energy in some of these environments and less food So animals start saving where they can. And one way is to start making less pigments, more gelatinous bodies, more water content, less muscle mass.
Yeah, probably
There's
Yeah.
the sea star there in the background. There.
Do you want to take a closer look at the sea star?
Yeah, let's try to get a good